All right. Well, hello, everyone, and thank you for joining today's webinar on mass spectral databases for untargeted exposomics. This is the 16th webinar in the exposomics webinar series for the Human Health Exposure Analysis Resource Program. My name is Chris Duncan, and I'm a program administrator at the National Institute of Environmental Health Sciences, and I'll be the moderator for today. It's my very much pleasure to introduce our speaker for today, Dr. Peter Dorstein. Dr. Dorstein is a professor in the Skaggs School of Pharmacy and Pharmaceutical Sciences in the departments of Pharmacology and Pediatrics at UC San Diego. He is a director of the Collaborative Mass Spectrometry Innovation Center, and he's also a co-director of the Institute for Metabolomics Medicine. Peter's research aims to develop new mass spectrometry-based methods to understand the chemistry of microbes, our microbiome, and their ecological niche. A few areas of his recent research focus include capturing mass spec knowledge to understand the microbiome, drug metabolism monitoring, the informatics of metabolomics, microbe-microbe and microbe-host interactions, and the co-development of the Global Natural Products Social Molecular Networking Knowledge Base, or GNPS. Today, Dr. Dorstein will discuss leveraging the high volume of public data generated from untargeted metabolomic and exposomic studies, and how analysis of these very rich data sets using repository scale approaches can give new impactful insights that are only observed if using these multiple studies altogether. And Peter will share insights on how mass spec data are indexed, organized, curated, and analyzed in large scale. And he'll also highlight some of the newer resources available and how they can be used to inform clinical cohort studies and determine influences on human health. We very much welcome the audience's questions today. So if you do have a question, please feel free to use the question and answer function on Zoom, and we'll do our best to address all of these after Peter's talk today. We also have the option to raise your hand, and we can unmute you after the talk as well, so feel free to do that at the conclusion of Peter's talk. With that in mind, let's move along to the presentation. Welcome, Peter. You have the floor.
Thank you so much. Let me pull up my slides real quick, and then thanks, Chris, for the introduction. Also, thank you everyone for joining the seminar. You can see that my title has changed a little bit, but what I'm going to try to do is show you the thought process behind how we can leverage these amazing resources that have been curated over the last decade almost with these public data repositories that exist, and how that could potentially be used to understand exposures and exposure science. But before I go on, I have some disclosures. I'm a scientific co-founder for Arome, Aveda, and Omeda, and then a scientific advisor for Cybele, Serenus, and Biomonitoring. I think most of you that are here, otherwise you probably wouldn't be joining this particular seminar, is that the exposure and the exposome has a big impact on the trajectory of health. And this could be both short-term exposure, acute exposure, as well as long-term exposures. And both of these are relevant to be able to assess so you can understand how that may influence health. But the real bottleneck is, and the real question that comes with it is, how do we detect different exposures? And this represents a little bit of a challenge. This is a challenge for anybody who's working in my laboratory as well. So if you, for example, if you want to say, I want to look to see if there are any antidepressants in human breast milk, we would then collect untargeted metabolomics data, in our case, LC, MSMS, where we fragment all the ions. And then we try to match it against this library. And so we have a reference spectra. If that matches to the data, we then assign that to be caffeine. That's just an example. And so in breast milk, it's a reasonable number of annotations that we may get is about 500. And so now we have this list of names that we get. And now the question is, well, which ones of these are antidepressants, or which ones of these are exposure molecules? You do a literature search, you go to Google, you go to Wikipedia, you use ChatGPT, and in the end, you have a list of all the molecules in this case that are antidepressant, or if you care about food-derived molecules. But this is a pretty manual process in general still. So now that person graduates or moves on, gets a faculty position, which is wonderful. But now we have a new person come in, and mass spectrometers are not experts in the molecules that they look at, particularly in untargeted metabolomics experiments. There's so many different molecules, they may have never seen them. And so if you, let's say, analyze a fecal data set, now you want to understand, is anybody taking antibiotics, and how does that impact the gut microbiome? You go through that same process. Maybe now we have 1,500 annotations, and we do the same process, and now we find some antibiotics. But it's inherently a non-systematic approach, and it doesn't capture the knowledge that the previous person gained. And so if we expand this into drugs, food, microbes, pesticides, plants, and we go through this sort of process each and every single time, you're really going to be spending a lot of time even just simply curating that metadata table to say, okay, this molecules from microbes, this molecules from food. And what's even more remarkable, if you take two people with the same list of molecules, you may not get the exact same source information. So you may not, somebody might assign a molecule to a microbe, whereas somebody else may assign it to a food, and in reality, they may be present in both. And so this represents one of the limitations in untargeted metabolomics that my laboratory faced over and over and over. And so the thought process that we had to make this easier is actually leveraging these large resources that have been created. One of those are these reference libraries. And the idea behind reference libraries, these are the known molecules. So you have some sort of structure associated with it. You have an annotation associated with it. But now we can tag essentially metadata to this library or reference library spectrum, so that when we encounter that particular spectrum, we don't only get the name of the annotation, but also the metadata, where it's a microbial molecule, it's a drug molecule, it's a pesticide, et cetera, et cetera. The other strategy is actually taking entire reference files. And so if you have, let's say, a tomato, you haven't done an LCM-SMS on a tomato, we then attach that metadata tag to it, and it captures both all the unknown spectra and the known spectra. So every single spectrum in those reference files are now have this metadata attached to it. And this represents a strategy to build a reusable approach to leveraging what's already available in the public domain. And the first example I'm going to show you is just one of the examples that we have for reference libraries. And it's really led by Nina, Kine, and Karina. And they were the drivers behind this particular project. And the idea was, largely, can we basically create a resource that allows you to quickly assess whether something is a drug or not? Or if you have a large cohort that you're studying, which samples that you studied actually contain different medications. So, but why do we want to really develop sort of this empirical strategy for medication exposure readout? Because in principle, we should have really good clinical records, and they are getting better and better and better. However, there are some limitations with medical records. Over-the-counter medications are not documented. We cannot assess adherence or compliance to the treatment regime. People are also super creative in getting medications. I mean, I could go 25 miles from my house, I can go across the Mexican border, and I can pretty much buy any medications I wish. You can order them on the dark web. We did that for a specific project together with law enforcement. And essentially, you can order any medications on the dark web. If you order antibiotics, you get free Viagra, for example. That was one of the perks that we found out when we ordered via the dark web. But also people are creative in using leftovers, either from people that live in their house, maybe a friend that has some extra medications that they didn't use and you're not feeling well. People do tend to grab some of this. However, there's also a lot of inadvertent exposure where you do not know that you're exposed to different medications. And that's because our food supply actually contains quite a lot of medications. The foods that we investigated so far, 20% of them contained some sort of antimicrobial agent. And generally people think, well, that's added to meat. No, it's often antifungals that are added to, let's say, fruits and vegetables, but also even organic yogurts or dairy products. They often contain, let's say, the antifungal natamycin, which is in humans used in fungal treatment of eye infections. But it's a product that can be added to organic dairy products to prevent fungal growth. And so there's a lot of exposure to medications through our food supply that we're not aware of. The other aspect of it is, okay, medical record, let's say you get a record from the last 2 weeks or the last day of when the sample was collected. But kinetics and distribution is really important. And so the sample that you're investigating may not have that particular drug in it, even though the clinical record says it has been taken because of the kinetics that exist. And so the idea that we want to, the idea behind it is really, if it takes 1 year to do this kind of work, can we get to it in an hour? And of course, we're not there yet, but you can see the progress and the direction that we're taking with the resources that we're building. And so the question is how? So first of all, we need to get a spectral library where we know that they're drugs. So that's the first thing. And then we attach a variety of different metadata that might be of interest to the community to quickly assess. So if you want to find out all the antibiotics that are present in the sample, you just find that infectious disease or antibiotic metadata tag and then filter for it. And so the reference library that we used are the open source reference library in the GMPS ecosystem that contains, at that time, about 600,000 MSMS spectra. It's now double. But essentially, we tried to see if any of the names or any of the structures contain in any of these sort of drug data bank resources that exist, and then in combination with expert curation of pharmacists. And in the end, the GMPS ecosystem of those 600,000 spectra, about 105,000 of these were MSMS spectra of drugs, representing about 5,000 different drugs. But now, if you really want to understand if somebody is exposed to a drug, you also have to know about the metabolism. So a lot of drugs are pretty rapidly metabolized, although there's individual variation in that. So some people may metabolize it very quickly, and other people may not metabolize it at all. But if the drug is metabolized in one person very quickly, you may never encounter the parent drug. And so, if you want to establish exposure, you need to be able to figure out the signatures for the metabolized versions of these drugs. And so the way we got to some of the metabolized reference spectra was essentially taking these 105,000 reference spectra, and basically we did analog searches, where we did analog searches against the entire public library. And the reason for this is, let's say you have this drug right here, it has a hydroxyl group and it gets methylated, or the other way around, demethylated. The signature itself should show up having a 14 Dalton difference. And so we can now basically say, okay, we're going to do an analog search with those 105,000 reference spectra against these public data sets that are present in the GMPS massive ecosystem. And nowadays, we can also do this against Metabolomics Workbench and Metabolites, the two other metabolomics repositories. But this library is created with just searching against the GMPS massive. And we do this with a mass spectrometry search tool, which we introduced in 2020. But essentially, we can do an analog search, as we show here. And the way to think about a mass is essentially very similar to the way BLAST works, where you can enter a protein sequence, DNA sequence, or RNA sequence. You can do BLAST with it, and you can find mutations, analogs of those sequences. So in mass, we can search with these MS2 spectra, and nowadays we can search across 4,300 public untargeted metabolomic data sets. But that essentially, when we did analog mass, that resulted in 1.5 million spectra that were similar to the drug spectra. And so then we filtered this based on common phase 1 and phase 2 metabolism. So the demethylation, for example, oxidation, dehydration, glucuronidation, etc. So we filtered for those, in at least the ones that we could explain. It's likely that many of the ones that are filtered out in this particular step are probably less studied modifications, so less studied metabolism. And that would be a research effort in the future to try to identify what those are. But for now, we kept phase 1 and phase 2 metabolism, resulting in about 250,000 reference spectra. But then we also filtered for ones that had library matches. So one of the things about drugs is that there are many drugs where basically they do medchem on and they find analogs of these drugs and they market the second generation of it. So lovastatin versus simvastatin is a one methyl group that is different. Here we have a match that we have one of those MSMS spectra. We then get a crinopril analog, and the mass difference between crinopril and this analog that we see from these spectra matches up to 2 carbons and 2 protons. So this, but then when we search the public library against the GMPS libraries, we find that it also matches to ramipril. And so this is where that C2H2 difference comes from. And so if we have a match to a known molecule, we then remove this analog match from this library as well. So that resulted in a total of 75,000 reference spectra that are really likely going to be metabolized versions of these drugs. And so in combination with the reference library itself, we have almost 200,000 reference spectra in this particular resource. And we have now linked this all to the drug metadata in terms of disease area, mode of action, drug names, and much of this was curated with these particular resources, but we created an ontology that could be readily interpreted. Now, then Kina, who's an excellent pharmacist, got together with a team of other pharmacists, and she further manually curated. And one of the challenges that we had is when we started to use this resource and test it against real data sets, we started to see hundreds of thousands of matches to taurocholic acid, for example. And that makes sense because this is an endogenous molecule. Or we started to get matches to lactinosol, or quite frequently. Again, because there are analogs that look very similar to that. And so we decided to add another layer of ontology to it, and that is based on the exposure source, because the endogenous metabolites you're going to, or drugs you're going to see quite often, and you wouldn't be able to differentiate those from being given as a pill or as an injectable versus what's endogenously produced. Whereas these sources that are of medical origin are actually, they're synthetically made in a laboratory. There are no matches to endogenous molecules. Those are much easier to interpret. So then the question is does the drug library do what we want it to do? And so to test this, we took a data set where it was known that people were given certain drugs. So here's a group of 10 individuals that were given diphenhydramine, and 10 individuals where they weren't given diphenhydramine. And then here was 13 people that were given caffeine, midazolam, and omeprazole. And what you will see is that in 10 out of the 10 people that were given diphenhydramine, we did detect diphenhydramine. And that's both in the plasma as well as the skin samples. However, there's a time dependency to this, and this is also shows why it's so important to complement clinical records, drug medication readout from clinical records, is because there's a really important time component to it. If you say, okay, we take people based on their clinical records at 24 hours, the drug may not be present in the sample that you're studying. So it's really important to get information on the sample that you're studying, because that will influence the rest of the metabolome and the rest of the biology that you're going to be looking at. So we can expand the question of, is the drug library reliable? We can go to actual data sets. So here's an HIV plasma data set and then a COVID plasma data set, and they should have very different medications that are present. That makes sense once you look at it. And so here, if we look at the HIV cohort, we see many of the HIV drugs that are not observed in the COVID patients. And then in the severe COVID patients, we tend to see antibiotics, flu drugs, and anti-inflammatories such as corticosteroids, etc. So we then decided to push this and think about it. Okay, what's the drug exposure in the general population? And we used this particular public data set from the American Gut Project. It's about 2,400 fecal samples. It's a citizen science project where you can basically pay money and you get your samples sequenced. And we also started to collect metabolomics data on this. And these are the different countries that I'm going to talk about in this particular project, but you see samples from the US, Australia, and the UK. And so if you now take this library and match it against the data that was collected for this particular project, this was all LCMS based, what you can see is that in 26% of the samples, we see matches to this drug library. And you can see in some cases, even up to 5 different drugs are present in a single individual. Totally, 30% of the individuals we found drugs in the USA, 16% in the UK, and 22% in the Australian samples. And because there's a lot of metadata associated with this American Gut, we can compare it to, let's say, self-reported age, as well as whether they're male or female. And you can see that erectile dysfunction drugs are only observed in males from the age to 30 to about 75. So that was quite satisfying to see. But also antihypertensive, antihyperlipidemics, you start to see those emerge around the age of 45 all the way up till very old age. And then all analgesics, antihistamines, antidepressants, antibiotics, they're more widely used throughout the entire lifespan. And so, you know, the NIH Institute that supported the creation of this library was NIA, and this was a part of a supplement of a U19 called the Alzheimer's Gut Microbiome Project. And so here we're trying to connect metabolomics features as well as exposure features to the responses that we're seeing. And in this case, we're looking at bile acids. So many of the bile acids, actually the majority of the bile acids that are detected, originate from the microbiome. So it's a good reporter of how the microbiome might be changing in relationship to the different drugs. And so if we look at just antibiotics that were found in this cohort of 635 individuals, we found 35 matches to antibiotics. And now taking this subset, we can really see how the bile acids metabolome is changing. And that basically, when there are no antibiotics present, the diversity of bile acids is much higher. Essentially, no new bile acids are observed when the patients are not treated with antibiotics. And we can do the same thing for proton pump inhibitors. If you want to go individual drugs, such as metformin, you can do that as well, or statins. And you can see that these all seem to influence the biome, so to speak. But then the drugs that are often seen in Alzheimer's patients, such as antidepressant, antihistamines, they had less of an effect, except in a few rare cases, on the differences and changes that we see in bile acids. So in this case, we see a tryptophan conjugates, which seems to be higher in the patients that have antihistamines, for example. So we're expanding this. So right now, I've only spoken about the drug library that we're curating, where we're adding these metadata tags. But the NIDDK supported us to create this for microbial metabolites as well. And so this is a part of a U24 that was funded. And up at this point, it has 1,600 microbial metabolites curated. And if you're interested in it, you can go to the website here. But so far, the majority, when you do untargeted metabolomics, the majority of the data remains unannotated. And so the real question is, in part, is how can we also leverage unannotated data to inform us about different exposures? And so we're going to take two strategies with this, or the thought process at least that we have is that we're taking two different strategies. One of those is metadata-driven molecular networking, and the other one is the creation of domain-specific masks. And I will get into details and explain both of these. But basically, metadata-driven molecular networking, I will just highlight what this is and how we do this, is basically, if you want to ask the question, if you have an untargeted metabolomics data set, is it possible to read dietary patterns from this? And the real question is, can we empirically establish diet from untargeted metabolomics data? And so the fundamental principle behind this is that every molecule in your body, well, at least 99.99% of what is in your body, has to be ingested. So it has to be either consumed from a meal or part of your beverage. Some of it will be from inhalation, some of it will be absorbed through the skin, but the majority of molecules that are in your body originate from your diet. And so if that holds true, that means that dietary signatures must be present in these clinical samples. And so if that's true, then we should be able to get some sort of diet information from the molecules that are detected in these particular samples. But again, the majority of molecules remain unannotated, particularly food-derived molecules, microbial-derived molecules. They are not yet annotated because people haven't investigated those sufficiently enough. And so the idea that we had is, well, what if we create reference data of food? So do LCMS-MS, untargeted metabolomics data on food, and or grab that reference data from the public domain, and then do co-analysis with clinical samples so that we can basically, in the end, get a food ontology out of it and some sort of spectral count in terms of the frequency with which those foods are observed. And based on that, you can begin to develop a proportional dietary pattern among the different individuals that you're investigating. The important part of this entire workflow is actually the food ontology, which is the largest curation project that has ever gone on in my laboratory. And that is because creating a food ontology is not that straightforward. And the idea was that we wanted to have a food ontology. So if we have an MSMS spectra, that we would be able to map it up which foods contain that particular MSMS spectra. But if I would have taken a strawberry, for example, I would have classified this as a berry, but a strawberry is actually a pseudocarb. And so if you did not know this, you would have classified it incorrectly. And so we've spent a lot of time, probably more than 2,000 person hours, on curating this particular ontology so it can be reused. And so now we apply this reference resource that we've created. We're going to be co-analyzing with clinical samples. And basically, there are some wonderful public clinical cohorts that are available. And the focus here is on IBD at this point. So when we do the co-analysis, we get this data table out of it. and then we can do PCA analysis on the resulting proportional dietary patterns that we were able to obtain. And when you do that, this is what you observe. So first of all, the circles represent individuals with Crohn's disease, whereas triangles are healthy controls and the squares are the ones that have ulcerative colitis. And then we color code this based on the ontology associated with fleshy fruit. So these are the spectral counts associated with fleshy fruit. So you can see the individuals on the left have more matches to fleshy fruit. The people on the left have more matches to vegetables. And then the people on the right have more matches to meat. And then the people on the top have more matches to fermented grapes. But once you have this data organized in this fashion, you can do regression analysis and you can see if any of these proportional dietary patterns match to some of the clinical information that you have available to you. And so when we do this, when we look at disease, and that means if it's ulcerative colitis, Crohn's disease, or non-IBD controls, you can see that dietary patterns, the proportional dietary patterns, or the spectral count based on spectral counts, are not statistically significantly changing between these different conditions, ulcerative colitis, Crohn's disease, and non-IBD controls. There's no apparent dietary patterns that exist within age or sex. However, if we look at microbial diversity based on sequencing the microbiome, now there's a higher diversity the more matches we have to fruits and vegetables. And that's something that has been documented in the literature, but it's really nice to be able to see this being pulled out of this particular data set. But now if we look at disease activity, and this is based on endoscopic scoring, the worse the disease was, the more matches we had to meat products. And then the other interesting observation was the more matches we had to fermented grapes, the less severe the disease or the fewer flare-ups that existed based on the endoscopic scores. And so you can eventually start to think about maybe starting a clinical trial. It would be a popular clinical trial where you add one of the components in this diet study, a wine component, to see if that can reduce these flare-ups. But the validation cohort, a small clinical trial that we're doing, is now being supported by the Crohn's and Colitis Foundation, the Helmsley Foundation, to see if we can validate this sort of metadata-driven metabolomics approach to see if we can guide this into a precision medicine strategy. And so that leads us to the second strategy that uses reference files to begin trying to assess what are exposure molecules or what are the source of the different molecules, and those are creation of domain-specific masks. In the area of my laboratory, as the introduction indicated, the area that my laboratory is really interested in is the microbiome. We're really wanting to be able to detect microbiome-derived molecules and ultimately understand their function. And so if you were to collect an untargeted metabolomics data set on a fecal sample, you get this big black sphere of data. If you were to provide annotations in 2016, the average annotation rate would have been 1.8%. Now this has grown to about 14 to 18%. But if you now go and do the Google searches, the literature searches on all the annotated metabolites, you'll find that we could annotate maybe at best 1%, if you're really lucky, 3% of the data to be assigned to be microbial. So it's a really small portion of the molecules that are annotatable can be assigned to be of microbial origin. And this is very different that if you were to take a metagenomic or collect metagenomic sequencing on a fecal sample or metatranscriptomic or do metatranscriptomic sequencing on a fecal sample, you can assign the majority of sequencing to belong to the microbiome and only a small portion to the human genome. So there's a disconnect between the number of molecules we can assign to be microbial versus the number of genes that we can assign to be of microbial origin. And so the idea that we're trying to work on is essentially, can we make it more efficient to figure out, even if the molecule is not annotated, to assess whether it's from food sources, other exposure sources, drug sources, but also of microbial origin. And the question is, can we leverage this public data that already exists, to make these assignments? And this is really how the start of the MicrobeMass, which is a domain-specific mass. And the idea was, we want to be able to assign known molecules to see which microbes can produce them, but also the unknown molecules be able to assign, or the unknown signatures that we get by mass spec, to be able to assign which microbes can make those. And so we recently published this, so it was pretty exciting. But the way we built MicrobeMass was essentially take public data that's in GMPS, curate the taxonomy, but we did not understand most of those data sets. So I put out some feelers in social media to see if you have data of microbes that have been grown in culture in the public domain, please reach out to us, because then we can taxonomically curate this so that we can make a good search engine. So in 2 months, we had about 100 scientists that reached out for this, and we curated more than 100 million MSMS spectra that have been observed in microbes grown in culture, so of bacteria, fungi, and we added human cells as sort of a control that, if this metabolite is observed in bacteria and fungi and in human cells, then it's probably not only produced by bacteria. And so, for example, tryptophan would be found in many of these. So to build a taxonomic tree, we have about 60,000 unique LCM-SMS files that have this curation being done by the community, resulting in about 2,300 NCBI IDs. And those fulfill about 2,800 taxonomic branches on this particular tree. And they really come broadly from bacteria, some archaea. We'd like to have more of these because those are important in the microbiome sphere, and eukaryotes. It's about 12,000 fungi. Almost all of the other ones are bacteria, and very few archaea. But then we coupled this with a fast mass search that we have implemented, and then combine it with this particular readout is what allowed MicrobeMass to be how MicrobeMass was developed. And so that allows you to basically say, okay, if I have an MSMS spectra and I search with MicrobeMass, so for example, phenylalanine cholic acid amidate, this is a microbial metabolite that we discovered in 2020, or we published in 2020. When you search with this particular MSMS, now you get this taxonomic tree out of it of where it's been detected. So in bifidobacteria and Enterococcus and some other organisms. And so this is a pretty powerful strategy of data reuse that allows you to then interpret at least microbial metabolites. And so we use this to extract putative microbial metabolites. And the strategy that we employed is, there was a public data set that we had from, I think, 2018 of germ-free mice colonized with a microbial community. And the first filter that we applied was basically, we kept all the MSMS spectra that were only observed in the mice colonized with a microbial community, because presumably those are more likely going to be microbial metabolites. And so we started with 1.9 million MSMS spectra, but 30,000 of those are unique. So there's a lot of duplication in these public data sets. And the filtering resulted in about 10,000 unique MSMS spectra. And then we searched against MicrobeMass to see if any of these could be produced by microbes in culture. And we found about 3,200 MSMS spectra that could be made or that were produced by these organisms. And then we removed any MSMS spectra that were also found in the human cell lines. And then we had a validation against a mouse study where they had treated mice with a normal microbial community with 5 different antibiotics to essentially eliminate the microbiome. And so once we filtered this, we were left with about 512 MSMS spectra. And here are sort of the taxonomic classifications of where these 512 spectra are observed. And so we took those 512 spectra and wanted to ask the question, well, do we see those spectra in humans at all? And so here we used masks, and we found that 455 of these were found in humans. And here we just have a map of health conditions versus organ distribution or biofluids that's available for each of the data sets, the human data sets that it would match to. And then here's just a summary of where we find these particular metabolites. So the majority of them will be found in stool, but even some of these microbial metabolites are found in the brain. They're found in saliva. They're found in breast milk. And so you can clearly start to see some evidence that a lot of these microbial metabolites end up in very distant organs. And we can repeat the same thing with cohorts of data sets that exist. So in this case, it's a sepsis study in small infants. And here we see microbial metabolites over time. Basically at the onset, they are not detected. And as the microbial community starts to take shape, we start to see these particular microbial metabolites. And this is now using micromasses pretty quickly to assess what is likely going to be a microbial metabolite or not. So micromass is one of the domain-specific mass we have. We have food mass based on the original ontology that we created, plant mass, drug mass based on the ontologies that I previously spoke about. But also we're adding entire formulations of drugs. So basically the entire pill that you're taking and that being extracted and run. And we have tissue mass and are slowly beginning to build an exposure mass. This is a UO1 that's funded by NIA as well. And here's some of the first hints of or examples that we're beginning to build out with this particular UO1. So in this UO1, we're starting to collect reference data on personal care products. We're starting to collect reference data on the entire formulation of drugs. Plastics, wood products, whatever people use on their daily basis, rubber from tires, etc. We're creating a reference data set. So far, we've really pushed the personal care product space a little bit more because we assumed that a lot of people would be exposed to them, and so it would be good to be able to observe those. So if you look, for example, this emulsifier, cyclodextrin, they're commonly in bath and body products, skincare products, so body washes, facial cleaners, lotion, moisturizers. And now if we look at the rest of the mass matches, we can see that they're primarily found on the skin of females. There's no other samples where this particular molecule has been detected. But the question is, do some of these molecules that are present in these sort of personal care products, do they end up in people? And so here's an antioxidant that is very commonly used in facial cleaners, lotions, skin care, perfumes, colognes. And in this particular case, what you will see is that they're found in blood. They're found in feces. They're found in the lungs. They're found in saliva. They're found in the vaginal tract. And this is based on matches in the public data. And so it would be really interesting to understand how does this antioxidant really affect the health trajectory of people that are exposed to it. But it's clear that this clearly gets systemically incorporated. And so the final example that I'm going to leave you with is our project on the International Space Station. It's basically a very unique and extreme microbial environment. This is a project with my co-worker, Rob Knight, and Rudolfo and Nina are the ones that are leading this particular project. And so this is a project together with JPL, these particular individuals. But the environment at the International Space Station in many ways is very unique compared to what you might find on Earth. First of all, there's weightlessness, there's more radiation, there's very limited nutrients. The only nutrients that exist is the food that is being brought onto the space station with these rockets that are being sent up. There's recycled air and water. There's a reduced hygiene. There's reduced amount of cleaning that exists because you cannot really bring that many towels. You cannot bring that many cleaning supplies up into the space station. And one thing that people have already documented is that there's an increase in antibiotic resistance, there's an increase in virulence, and there's a decrease in crew immune function. And so one of the goals, as we're starting to explore going to Mars or even other distant places, is to identify and mitigate risks of human health exposure during space exploration. And this is really, instrumental as we start a habitat to space. But one of the challenges that you face when you start to do these sort of space projects is that the best tech that you have on Earth, and in our case, we used a lot of these swabs that have ethanol in it, but these are these 15 centimeter tubes. That if you send things up in rockets, you don't want to take up a lot of volume. We also couldn't send up anything with ethanol. And so we needed to design our own tubes that were less than 5 centimeter in size, but still had these dual cotton swabs. And so we did 3D printing for this. And all this planning started in 2019. We went to the JPL National Laboratory, went to Houston, to look at the mock-up for the space station of how this would really go and be done. And so in December 2020, these samples were launched. This is the rocket that was going up and that eventually delivered this cargo to the space station. And then in 2021, the sampling happened, and in totally, Kate Rubis, which was the astronaut that was doing the sampling, she spent 14 days of sampling. She got 803 samples total, but she took pictures of everything she sampled. She really documented every location of where in the space station this was sampled. In total, we were able to get samples from 9 different modules, and then those were sent back to us. And so based on the map that the astronaut created, we have this 3D image where each of these circles represent the location of where the sampling was taking place. And here we're actually looking at the toilet and exercise area. So the toilet is on the left, the exercise area is on the right. And so when we performed LC-MS analysis after blank subtraction, we can start to see about 3,200 mass spec features. And you can start to see that some of these are very uniquely observed in just one module, and then others tend to be distributed pretty widely among all the different modules in the space station. And so one of the kind of molecules that we saw were based on disinfectants. And yeah, you would expect those to be around the toilet area where they would want to do some cleaning. But if you start to look around the toilet area, you can also start to see, well, there's no real ceiling there, but on the bottom or the left, on the right, on the top, above, you start to see these quaternary ammonium compounds that are part of this disinfectant wipes that they have available on the space station. We also start to see evidence for microbial metabolism. So here are urinary metabolites or fecal metabolites also. And you can indeed see those along the toilet. They're commonly observed there. And then if you take the sequencing data, you can indeed see that there's quite a lot of different gut microbes that are present around this region, but not only the toilet area, but also all the areas around the toilet. So just cleaning that local area does not eliminate the presence of fecal and urine metabolites throughout the entire module. We could start to see other exposure molecules, so industrial chemicals, so sealants materials. And this was largely observed around these rubber mats that are part of the exercise equipment, where they put these rubber pads on them so that preventing them from slipping. And they actually use the sealant shown right here. And that's one of the things that we detect. We also see these forever chemicals, but this was only in the module, exercise module, and right across from it, they had a plastic sheet. And this contained a lot of these PFAS molecules. And then we start to see pepper-derived molecules or food-derived molecules on the dining table, as well as the food storage modules, and even where the food processing is taking place. But in the same region, we also found fungal-derived metabolites. So clearly that some of the food particles that have remained in this module eventually will be inhabited by fungi that will start to metabolize those as well. However, what I've described so far has only been based on the annotated molecules. It's about 3%, 97% remains unannotated. And so how can we better interpret this? Well, I just described to you that we have these tools now, such as micromass, food mass. And when we apply this, we can assign 14% of these molecules that are detected to be of microbial origin. Only a relatively small portion, maybe 2% or so, could be assigned to food. But that's because the reference data that we have only contains food that we consume on Earth, and it's only about 3,600 different foods, and there's literally hundreds of thousands of different foods. And so ideally what we would have is reference data from the food that astronauts consume. And my suspicion is that a larger percentage of these molecules will be assigned to be of food origin. So one of the questions that we have is how similar is the ISS space station metabolome to the metabolomes that might be observed in human habitats on Earth? And so we have data on urbanization gradients. And so from the jungle all the way to the city. And we basically, now it's possible to do co-analysis and ask the question is, okay, what molecules or what signatures are missing in the space station that might not be present, that might be present on Earth, or what molecules do they have in common? And so we start to look at the rural metabolites, and what we found was that there's a lot of flavonoids, lactones, a lot of plant-derived molecules are missing in the ISS space station, whereas those are readily observed in the rural area. And then there's some molecules that might be coming from food, such as hydroxy fatty acids and terpenoids, that are observed in the space station as well as in rural towns. And then a lot of molecules are observed only in cities and ISS space station. And this is largely these molecules that are part of our cleaning habits, such as the quaternary ammonium salts, cocoa beta-betaines that are part of soaps and shampoos, etc. So what we're learning basically is that the ISS space station is most similar probably to urban homes, but that there's also a lot of missing molecules. And the question is, do those missing molecules influence health? And this is something that NASA will have to sort out. And if they do contribute to human health, how do they can then introduce those during long space travel? So in summary, I've shown you sort of the thought process and the resources that we're starting to build of reference libraries that can be used to understand exposure, use in untargeted metabolomics data. And we have both reference libraries where metadata is attached, as well as entire reference files where the metadata is attached. And so what we can now do is not only provide structural annotations of the metabolome when you do untargeted metabolomics, but also provide source information. And with that, I want to thank you for your attention. This is the team that really does all the work. I've highlighted many of them throughout the entire talk. But this work is not done in isolation. Most of these people still collaborate even when they've moved on to their own faculty position. I think Justin Vanderhoofd is listening in. He's one of them. But there's also about 1,500 co-authors and collaborators in the last few years that have really enabled this science that I just presented. And then finally, I will take any questions you might have.
Thank you so much, Peter. What a fascinating talk. Yeah, yes, as Peter mentioned, with the remainder of our time, we got a few minutes to take any questions. So as a reminder, please feel free to add your questions to the Q&A box, or if you prefer, feel free to raise your hand and we can unmute you. We do have one question, Peter, in the Q&A box right now, and it's from Chase Clark. Thanks for the great talk. With the food omics, have you used spectral similarity to look at healthy versus disease group differences in the products of microbial fermentation or metabolism of the ingested food compounds?
So I think this is a great question. So we took initially, so when you do co-analysis of the food and the clinical samples, so we took a brute force approach, and we initially primarily focused on what was only identical. However, Chase is right in the fact that a lot of these metabolites undergo metabolism. And so one of the things that we have done, but we haven't done this routinely, is in the molecular networking, you can look for analogs of these metabolites and also assign those to belong to those particular food categories. But it is difficult. If you were to consume grapes and consume wine, essentially the process of making wine from grapes is a metabolism result. And so it is actually pretty tricky. And I think we will still need to develop new technologies that would allow you to differentiate in a very rigorous fashion whether somebody consumed grapes or whether these were wine products. So I think so. Molecular networking is one way that you can begin to propagate. I think there are some other creative ways where people can assess metabolism, but those have just not been employed yet. So if anybody is interested in developing those resources, I would love to work with them.
Great, thanks, Peter. And we'll take one more. We've got a question from Justin Vanderhooft. And again, thank you for your great talk, Peter. Do you plan to extend the food mass principle for the reference-based metabolomics? And if so, what is your approach to grow it?
Yeah, so that's a great question. So we're primarily biased with the foods that we have that were really accessible to us. So primarily what students and faculty around us were eating. That's likely also not representative of the entire world population. So there's two strategies that we really like to use to expand the food reference library. One of those is reaching out to different people in different communities so that we can get those samples and run them. The other one is collaborate with people who have an interest in food analysis and working with them that they also collect data on those so that we can really expand the reference library of food. I think this is one of the necessities. And we've seen this also, that if you take a Hispanic community versus a non-Hispanic community, and you don't have reference data from that Hispanic community, you don't get as many food matches than if you do include the reference data from the Hispanic community. So there's real value of getting more. The more data you have, essentially, the better it will be. And once you have enough data, I suspect there are going to be some really good machine learning strategies as opposed to the brute force approach of spectral matching we use now to be able to assign the food composition based on these based on clinical or matches to clinical samples.
Thank you, Peter. And looks like we're out of time for today, but in closing, just want to thank you, Peter, for your excellent presentation and thoughtful insights. And I'd also like to thank the HERE Network and the organizers of this HERE Exposomic webinar series for the opportunity to highlight this very important topic today. Before we sign off, I'd like to share one quick announcement. The next webinar in the Here Exposomics webinar series will be on Wednesday, November 20th from 3 to 4 Eastern. So please feel free to join that and certainly encouraged to join that. The speaker will be Dr. Itai Klug, and he'll be discussing how we can leverage resources in the satellite and geospatial fields to generate external exposome data sets to address the effects of climate change on human health. For more information on this series, including recordings of today and past webinars, please visit hereprogram.org. And with that, I'd like to say thank you again to Dr. Dorestein, and thank you to everyone in the audience for joining us today. And we'll see you in November.